Okay, thank you. Thank you very much for those beautiful stories. I really hope that a lot of people are going to sign up and be mentors and mentees. That definitely helps. So we are now going to move into our next session for today, the day number 3. This is a really interesting session that I really hope that brings up quite a lot of discussion. So for this session, we're going to have 2 chairs. The topic of the sessions are challenges and solutions from a regulatory perspective. One of the chairs is Juan Vale, who needs no introduction. Please, Juan, join us here. And the second chair is Professor Jennifer Knox. So Dr. Jennifer Knox is a professor of medicine at the University of Toronto, is working as a staff medical oncologist and researcher since 2001 at Princess Margaret Cancer Center, is currently co-director of the McCain Center for Pancreatic Cancer. And as you probably know, her research interest includes multidisciplinary treatment and translational trials in pancreas, biliary, and hepatocellular carcinoma. She has been the study chair of multiple clinical trials conducted within the Canadian Clinical Trials Group, but also within the U.S. National Cancer Institute. A lot of these trials are investigator-initiated trials, but there are also trials with pharma companies, and is also a past co-chair of the NCI Hepatobiliary Cancer Task Force. Her significant career has been awarded, including the Wilfred Lewis Chair in Pancreatic Cancer Research in 2017, and in 2019, the Division Research Achievement Award also. And as we all probably know, Dr. Knox is sought internationally, really, to speak her research in oncology and leadership, and it's a pleasure to have her here today. Thank you both very much for chairing this session.
Thank you, Dr. Lamarca. We are really enthusiastic about this session. So we're going to continue the theme of challenges and solutions. You heard some of this yesterday when clinicians and investigators were talking about challenges and solutions in their countries. But we know that there are also challenges, and we hope to find solutions when it comes to regulatory processes and drug approvals. And I'm hugely grateful, and I think as a community we're hugely grateful to be able to welcome here global regulators to just talk through with us some of the examples that they've had in terms of challenges, but also how they've provided solutions in terms of drug approvals. So we're going to start off in China with Dr. Xiaoyan Chen, who is a professor and office director of the Clinical Research Institute of the School of Medicine at Tsinghua University, Beijing, in China since February 2019. Dr. Chen served as a senior clinical reviewer in the Center for Drug Evaluation in National Medicine Products Administration, that's NMPA, previously the CFDA in China, before her move to Tsinghua University. Dr. Chen's research focus on drug regulatory science and new drug development and evaluation, especially for clinical trial design and development strategies for oncology and hematology drugs. She served as the project manager for drafting several guidelines during 2005 to 2019, such as guidance for clinical study and evaluation of anti-cancer drugs and the guidance on clinical trial endpoints for the approval of cancer drugs. We'd like to welcome Dr. Chen.
Good morning, everyone. Before my presentation, I would like to say thank Professor Zheng invited me to attend this wonderful meeting. It's really a good opportunity for me because I was a clinical reviewer. I think it's really a good opportunity for me to communicate and learn experience from the other country regulatories. Today, I would like to give you a brief introduction about the rare disease development in China. I will show you some data about the trends policy and the guideline and some real cases as well. In the early of this year, my team had done data analysis about the rare disease drug development, both in global and China, which published on the Nature Review Drug Discovery. In this picture, you can see, as you can see, in the past decade, the China has already increased rare disease drug development, although it is still less than the global, but it really increased because of the regulatory reform since 2015. I will explain it a little bit later. In terms of the therapeutic area distribution, you can say the oncology drug remains the most active. While in China, the proportion of oncology drug is 71%, more than that in the global, which is the 43%. It should be noted that some tumor types is regarded as the rare disease in Western country, but not in China, such as the liver cancer, gastric cancer, and esophageal cancer, because there exists a significant difference of incidence rate. So I think this may give us some opportunity to develop a rare disease drug in global way. This picture shows you more detailed information about the tumor type distribution. Here, the purple one. The purple one is about the carcinoma in development. Yes, in the past of years, more and more compounds invested in this area. Even it is a really small indication, so you can imagine how competitive in this area. Yeah. I should explain more. The top three target. The top three target is FGFR, PD-1, and VEGF. Now I'd like to show you some policy and guideline in China issued in the recent years. Actually, the China NMPA always encouraged the rare disease drug development. Since 2015, there was a regulatory reform to encourage innovation. A series of policy, regulation, and guidelines were issued to encourage the rare disease drug development and innovative drug development, such as the expedited program and tax incentives, market exclusively, and a single-arm study, decentralized clinical trial, and so on. But I'm not going to the detailed information, all the regulation. I just focus on the clinical development guideline. In this guideline of the clinical development for rare disease drug, the guideline recommend a different way to develop those drugs. It means the multiple stage design was recommended by this guideline. It means at the first stage, you can conduct a clinical trial in a small group to give the proof of concept. Then go to the second stage to as a pivotal study to to support the conditional approval, then conduct the post-marketing confirmatory study. It's now most of Chinese domestic drug, oncology drug has been approved, via this pathway. Also, this guideline encouraged some complex design, such as the basket design and the umbrella design, platform trial, adaptive design, and so on. China also set up some expedited program as the same as US FDA. For example, priority review, conditional approval, breakthrough therapy designation. Also, I have to note that this urgent need overseas drug. We call it UNOD procedure. This procedure only for those drugs which have already approved in the other country and with highly unmet medical need. If there's no ethnic difference, it may be acceptable based on the overseas data. I mean, they can give the exemption without Chinese data, without Chinese clinical data, then approve it in China. All those pathways emphasize on the clinical value-based and covering the rare disease drug. This slide also shows your data analysis conducted by my team. We just conclude all the approved by China NMPA, all the approved oncology drug by the China NMPA. Between 2018 and 2022, China NMPA had approved 81 new oncology indication for Chinese domestic drug. Yes, it's many. Yeah, of them, 42% were approved based on the single-arm study with ORR, objective response rate or complete response as the primary endpoint. And most of them are rare disease, or rare tumor disease. And the sample size for those approved drug, both those approved single-arm study, the median sample size is 102, ranged from 28 to 100. The sample size for the rare disease is even below 100. As you see in the left picture, the red dot means the history control figure. The green one means the target threshold. Normally, we calculate the sample size based on the difference between those two figures. Here, give you an example. Pemigatinib. I'm sure you already know it very well. I just emphasize one point. For this drug, it was licensed in and co-development by China Innovent company, which is a domestic company. At that time, they are required by the China NMPA to do a bridging study to support this drug approved in China. The study is a single-arm study and with OR as a primary endpoint. The sample size is 30. Finally, the study met the pre-specified threshold and determined the consistency with the global data. It's a bridging study example. Now give you a basket design example. It's for entrectinib, which is a CHK and ROS1 kinase inhibitor. Both China and FDA already also approved 2 indications, include NTRK solid tumor and ROS1 non-small cell lung cancer. They are approved based on pool analysis data from the 3 single-arm study. They pull all the data, then supported approved by those 2 regulatory. China take part in one of them. We call it STAR-CHK2 study. It is a basket design study and covering different the target. I think it's really a smart design to support 2 indications approved in 2 countries. This is MRCT, a simultaneous MRCT example. Next example is about denosumab for the GCTB. The GCTB is a rare disease, and there's no available therapy at that time, and with highly unmet medical need. For this drug, we approved it without Chinese data, only based on the other country, based on the data from other country, using the UNOD procedure, as I mentioned before. And also another point I want to say that for this drug, they also develop a non-rare disease in China. So the data from non-rare disease also provide more safety data to support the approval in China. The last example, I want to here show you a Chinese investigator initiate a study. It is a umbrella design phase 2 study in lung cancer. The PI, Professor Yilong Wu, who is a famous oncologist in lung cancer, he designed this study in patient-centric design. As you see, this study includes 3 arms. The first arm is for the patient who are completely eligible to the inclusion criteria, and the second one, they recruit the patient who are not completely fit the inclusion criteria. For those 2 arms, they use the HER2 inhibitor to treat them. The third arm is real world group. It means the patient can use the physician choice to treat them. So it provides the real world data as a control group. Finally, both the first one and the second one show high response, while the real world group there's no response. So I think this example is a really good example to give us a new thinking how to develop a rare disease with real world control group. This is my last slides. I want to say the rare disease drug development is not only one country's problem, but the global challenge. So it's really important to set up some platform or a system to promote the collaboration between different countries and involve all the stakeholders worldwide. We need more collaboration and sharing resources to facilitate the rare disease drug development. So hope today's meeting, later discussion can bring us more new thinking, new solution to deal with this problem. Thank you for your attention.
Thank you very much, Dr. Chen. We're going to move on to Europe now. Next speaker, Paolo Foggi, should be on line. He was not able to join us. I'm just checking that we do have him available. Good morning, Dr. Foggi. Dr. Foggi is an oncologist with extensive experience in clinical research and drug assessment. After his tenure as a clinical researcher at the Regina Elena National Cancer Institute in Rome, Italy, he transitioned to the Italian Medicines Agency in 2010. He served as a clinical assessor for oncology products, specializing in clinical trial applications, scientific advice, and marketing authorization of centralized procedures. We welcome you, Dr. Foggi. I understand that you are at an EMA meeting also, so you were not able to be with us in person, but we're looking forward to your presentation. Thank you.
Okay, thank you. Thank you very much. I hope you can hear me well and see the slides. Please, if you can just confirm that so that I move on.
Yes, we can see you and hear you well. Thank you. Okay,
very good. So, yeah, thanks indeed for this invitation. I was really particularly happy to join you because I know the kind of huge work you are doing, the impressive work you are doing as a community, including very important and academics and together with patients. So I guess it's really an opportunity for me to be here and discuss also with other colleagues from other regulatory agencies. First, it's a public declaration of interest and more importantly, a disclosure that the view that you will hear is not to be attributed to my agency, Italian agency, or the European Medicine Agency. So what I'm going to share with you, it's first of all, I will go through a few slides just to give you a background and essential infos on the legal framework and requirement for marketing authorization in EU, just because I understand that the audience is mixed, and so I guess that not everyone might be aware that, and I guess it might help in understanding the message that I want to convey. And then I will go through a couple of examples, same condition, different approaches, to start a discussion and reflection. Then I will end with some reflection, and then I look forward to the discussion that we will have. So in terms of the legal requirements, it should be clear to everyone that a new marketing authorization may be granted only if the risk-benefit balance is favorable. That means that the benefits must outweigh the risks. Otherwise, it's not possible to approve a new medicine in EU. And it is true even in the situations that are specifically dedicated to the very rare diseases or situation in which there is a clear unmet need. Because in the EU legal framework, there is the provision for conditional marketing authorization. That means that you, in case there is an unmet medical need, you could approve a drug even if the clinical data are not comprehensive, provided that the risk-benefit balance is positive, as I mentioned before, and it is likely that comprehensive data can be provided in a second moment, and the unmet medical need needs to be fulfilled. That means sometimes that you need to demonstrate a major therapeutic advantage over the treatment alternatives that are already available. Then we have also this option of the under exceptional circumstances that are really intended for the indication that are really super ultra rare, in which it is really unexpected that the developer could be in the position to provide comprehensive evidence. But anyway, even here, based on the positive data that we might have, the benefit risk balance need to be positive. So when we think about the benefit risk balance at the EU level, we are talking about an absolute benefit risk in which we weight benefits with risks. And so the benefits are clearly on the clinical outcomes, and the risk are mainly related to the safety profile. Then even in the best of the cases, but especially in those cases in which we approve drugs based on the less than comprehensive data, we have to deal with uncertainty. Uncertainty very often when we are ready to approve are then addressed in the post-approval phase. And so what kind of post-approval measure do we have? We can, to simplify, distinguish between specific obligation, and these are conditions that needs are mandatory for the marketing authorization holders. And then we have recommendations that are important to complement the unknowns and the uncertainty that I mentioned before, but that are not mandatory. And typically in the conditional marketing authorization, we have a specific obligation that are mandatory and are required to convert the conditional marketing authorization into a full approval authorization. Otherwise, it might even result, if the results are not positive, then that the drug is removed from the market. And examples of a post-approval measure include post-approval efficacy study, safety study, or registers of drug utilization, just to give you a few examples here. So that said, just to drive you into the EU regulatory framework, I was requested to provide some example, and then I guess it was interesting to have these two examples, and I decided to go with two medicines that are approved for the cholangiocarcinoma indication, because I hope that also those that are not clinicians or industry or something more professional, but even patients can more easily understand, because they likely know very well what I'm talking about. So I have these two examples. I have the ivosidenib, which is an inhibitor, the mutant IDH1 enzyme, and the FGFR inhibitor instead, which is the pemigatinib. That were targeting similar indication, although of course they were targeting the specific population with the mutation. And you can see here that in terms of prevalence of the mutation in the whole cholangiocarcinoma, it's the numbers are overall quite similar, so not dramatically different, and especially in the intrahepatic cholangiocarcinomas, rare in the other subsites. But yeah, it's interesting here to look at what kind of dossier we were faced with. And it might fair to say also that we had the opportunity to discuss with the applicants in advance of the, while they were conducting the clinical development, they proposed the clinical studies that they would have then submitted. So if you look at the ivosidenib, we have a randomized phase 3 trial versus placebo. Crossover was allowed, randomization was 2-to-1, and the inclusion criteria are at least one, but no more than 2 prior treatments that containing a gemcitabine or 5-FU regimens. And as expected for a randomized trial, the primary endpoint here was PFS by independent review committee with secondary endpoint OS. I would say that it's quite a standard approach, regardless the fact that, yeah, we are in a relatively rare setting, but yeah, it provided to be feasible. Of course, we had some discussion with the applicants regarding, and it's all transparent and public, so I'm not disclosing anything not known already. We had some discussion on whether we would have preferred to see OS, and then we would have preferred to see an active comparator, but yeah, that's fine. That's a randomized trial, which is something good and provide to be feasible. On the other hand, we had met pemigatinib, and they came with the 5202, which was mentioned also by the Chinese colleague. And yeah, here the primary endpoint is expected for a single arm trial was overall response rate with a key secondary endpoint to duration of response. Now, just to what I want to flag here, if you look more or less, no dramatic differences. Of course, I'm not going into the details, just to flag that it was possible to enroll 185 subjects in the ivosidenib trial, and that resulted in around 124 patients that received the ivosidenib. And remember, the crossover was allowed in this trial. And then instead we had around 100 patients enrolled in the single arm trial for the pemigatinib. Just to and the enrollment period was around the same, or more or less it was feasible for both to enroll more than 100 patients in these trials. Okay, looking at the results, and of course, for the next slide, please focus on the left part of the slides on the ivosidenib, just to remind you the results that we had in the median PFS was not that outstanding, but if you look as a ratio, and if you look the PFS rate at 6-12 months, perhaps it's interesting, especially, and if you can look at the Kaplan-Meier curves of the PFS. And then we have some data on the overall survival. I remind you again that we had the crossover here, but nevertheless, we observed an interesting trend in median OS with another ratio of 0.79, which was not clinically meaningful. Now look at the pemigatinib. The pemigatinib, it was on the about the over, and no, I guess that I have just to show you the Kaplan-Meier curves of overall survival, almost superimposable, but with the trend to an improvement. Regardless, it's not perhaps so huge, but might be meaningful and make difference for patients. Pemigatinib of single arm trial, and then we have an overall response rate of 37% with 3% around the complete responses, with a meaningful median duration of response of around 9 months. So what I want to flag you to your attention, and perhaps it's the kind of explanation of why we were faced with these two different approaches. And if you look at the overall response rate for ivosidenib, it's needless to say that if they would have come with a single arm trial, this would never have fly, because with the 2%, it's really impossible to make any informed decision with a single arm trial. The situation was different when it comes to pemigatinib, because you cannot think that a response would have occurred without without the drug. So in a way, you can interpret that. Then we will see that it remains a lot of uncertainty when it comes to contextualize this kind of result, especially if you are looking at a subpopulation of biomarker-driven subpopulation in which you really do not know what the outcome and the prognosis and the responsiveness to treatment it is, which is the challenge that we always have with with the single arm trial. And of course, it's less than an issue if you look at overall response rate clearly outstanding. I mean, I'm talking about 80, 70, 80, 90% in a situation in which you would not expect to see more than 10%. But when you have a more borderline, that it might be more difficult. And but what happened there? It happened that, for ivosidenib, the marketing authorization application for cholangiocarcinoma was submitted together with the acute myeloid leukemia indication. It turned out to be a full marketing authorization by consensus from the CHMP, and the main conclusion in the benefit risk assessment is that the study results have shown efficacy in terms of reduction of risk of disease progression and death and durability of stable disease, safety profile acceptably characterized and manageable, with recommendation implemented to minimize the risk of QT prolongation, which was an issue, of course, that emerged during the development. And of course, the poor prognosis of the disease, the limited treatment option, and the high medical need was noted. But then it resulted in a full marketing authorization. When you look at pemigatinib, based on this single arm trial, there was a conditional marketing authorization that was granted, and it was not by consensus. There were divergent opinions. For people that was the opinion that the overall response rate observed was not outstanding. It was difficult to judge because, essentially we were not pretty sure of the meaning of from the prognostic on the responsiveness to our treatment on the being FGFR positive, and also it was weighted against the safety profile of the drug. And we know that, the toxicity of this drug is not negligible, although manageable. But anyways, regardless the fact that we have divergent, the majority of the CHMP was positive to grant a conditional marketing authorization because they considered that the results were clinically meaningful and durable activity, which was deemed high compared to the currently used therapies. The safety profile, although with metabolic disorders, gastrointestinal disorders, skin, subcutaneous, and not to mention the ocular toxicity and the unknowns about the ocular toxicity. But anyway, it was considered sufficiently characterized, although it was a conditional marketing authorization. That means that there are specific obligations for the marketing authorization holder to conduct and to confirm studies to confirm efficacy and safety post-approval. So this specific obligation in this case was to provide the final results of the 5202, the phase 2 trial that was pivotal for this indication, and the results of the 5302, which is the phase 3 of pemigatinib versus gemcitabine plus cisplatin chemotherapy. So interesting, a different setting and different line of treatment. That's it. So this is what we have seen, and I wanted to start from that, just to state clearly that anyway, randomized controlled trials continue to be the standard for providing confirmatory evidence of efficacy on new treatments. And a randomized clinical trial, whenever feasible, is expected. And the reason is that establishing efficacy based on non-randomized studies might be particularly challenging for reasons that I don't think I need to explain to you, lack of features that are instrumental to avoid bias, the absence of a control arm, and the subsequent need to rely on the external information to interpret the results. And then we have these issues that we often encountered and turn out to be a problem, and it's something important for I guess for the patients, for the entire community, and the challenges in having confirmatory comprehensive data to convert from CMA to a full MAD, not always feasible or provided despite the specific obligation, and it's an issue. Nevertheless, I want to flag you that I don't want to give you the impression that we are conservative, because we are not conservative at all. And we are very open to explore new routes. And also, if you look at the period between 2012 and 2021, you can see that 1/3 of the anti-cancer products were approved based on single-arm trials. So it's not that single-arm trials is something that we are refusing at all. But for sure, I guess that we can all easily agree that it's not the best way forward in the interest of patients, in the interest of good science. So, as I show you, there are plenty of examples of successful application based on SARS. Resorting to no-randomized trials is often justified in rare cancer. However, as we have seen, the acceptability of a single-arm trial and its primary endpoint is dependent on not only on the clinical context and the feasibility of a randomized, but also the mechanism of action of the drug. Imagine that with the drug, with the mechanism of action of ivosidenib, and the kind of lack of over response rate, it would be impossible to come and approve this drug based on a single arm trial. And I just want to flag you that we have put forward a reflection paper on the use of single-arm trials, pivotal evidence for marketing authorization, which has been published last year and was made for consultation, and now it's under revision. So even in the context of rare disease, it remains that even not sufficiently powered randomized study might be preferable over single-arm trials. They might allow obtaining unbiased treatment effect and comparative safety data, which are important from very important to I would say in the interest of patients over time. And what could make the difference in the next year? And I'm happy that I very much like the last examples that my Chinese colleagues made about the use of real-world data. High-quality real-world data, high-quality registries might be the key in the future to widening the design space for clinical trial in rare cancer, allowing the best use of external data and their incorporation in clinical development. That's something that we should aim to do all together, because there might be ways to use real-world data, high-quality real-world data, and try to shape new clinical development while maintaining the same high level of scientific rigor associated with the standard and traditional randomized clinical trial. So this was to start the discussion, and the real-world data, and something in which at the EMA level, at EU level, we are investing quite a lot, because really we think that it's key and could make the difference. And with that, I conclude my presentation, and I really thank you for listening to me.
Thank you very much, Dr. Fogi. And we're going to move on to our next speaker. This is now, we're in Japan, with Dr. Takahiro Nonaka, who is our professor in the Department of Clinical Medical Science and Medical Science at the Graduate School of Medicine and School of Medicine, respectively. Specializing in life science, medical technology assessment, and medical management, he's affiliated with the Institute of Medicine, contributing significantly to medical research and education. Dr. Nonaka.
Thank you for kind introduction. I would like to express my thanks for inviting me to speak here today. I'm Takahiro Nonaka. Before, until 2 years ago, I was a review director of oncology drugs review team at the Japanese Regulatory Authority, PMDA. I was involved in the approval review of anti-cancer drugs for about 12 years at PMDA. during which time I was involved in the revision of guidelines for clinical evaluation of anti-cancer drugs and the promotion of the use of real-world data in the clinical evaluation of pharmaceutical products. So today, I would like to talk based on my experience of working in the review of anti-cancer drugs in Japan. So I'm going to talk about the recent challenge and current situation in Japan. One is regarding on the precision medicine in oncology, and the other is accelerating global development on anti-cancer drugs. So to be more specific, I will introduce the guideline for clinical evaluation of anti-cancer drugs in Japan. And I will also introduce the Sakigake designation system and the conditional early approval system for pharmaceuticals. So the number of patients with individual rare cancers is small. However, rare cancers is collectively account for 55% all of cancer in Japan. Many people may develop a rare cancer during their lifetime, but the development of anti-cancer drugs for rare cancer is not easy, mainly for some reasons. First, because of small patient populations, recruitment for cancer trial is difficult, and pharmaceutical companies may not see a return on investment. Second, because of conducting a clinical trial requires a large number of participating sites and take a long time, high cost of drug development. Third, available biomarkers are limited. To begin with, since the 1990s, Japan has had an orphan drug designation system. The number of patients, medical needs, and the possibility of development are considered as a criteria for orphan designations. The criteria regarding the number of patients is less than 50,000 in Japan. So therefore, for example, 1/3 of the anti-cancer drugs approved in Japan between 2004 and 2018 were approved after being designated as orphan drugs. This also suggests that there is an unmet medical need for rare cancers. Now, looking at the anti-cancer drugs approved in recent years, for example, in fiscal year 2021, of the 23 anti-cancer drugs approved for rare cancer indications, 12 were approved with orphan drug designation, 11 of which were approved based on the result of phase 2 single-arm studies. In a similar trend in fiscal year 2022. So next, I would like to describe one of the major challenges in recent years in Japan, that is the revision of guidelines for clinical evaluation for anti-cancer drugs. The first edition of the guideline was published in 1991. It was then revised in 2006, and then in March 2021, making this the latest edition. So the latest edition for the first time mentioned the clinical evaluation of anti-cancer drugs targeting rare cancers and rare subtypes of cancer. As a method of clinical evaluation for cancers with small numbers of patients, single-arm phase 2 trial using response rate as the primary endpoint mentioned. In addition, the utilization of real-world data such as disease registries and tumor agnostic studies using biomarkers are also mentioned. This slide shows anti-cancer drugs approved for tumor-agnostic indications in Japan. So, in this slide, I would like to explain the Sakigake designation system for drugs. The Sakigake designation system was launched in Japan as an attempt to facilitate the development of innovative drugs. This system is similar to breakthrough therapy in the US and PRIME in Europe. There are 13 new drugs approved under the Sakigake designation system so far. So about half of them are anti-cancer drugs. The designation as a Sakigake also allows for priority regulatory consultation during drug development. It also allows the initiation of an approval review of study result already obtained prior to the filing of a formal application for approval. As a result, the review period required for approval can be reduced by more than half. This slide shows anti-cancer drugs that have been designated and approved under Sakigake designation system. Six drugs have already been approved. So next, I would like to explain the conditional early approval system for drugs. This system is applicable to drugs that are eligible for priority review, for which it is difficult to conduct confirmatory clinical trials, or those that will require a very long period due to the small number of patients, and for which determined efficacy and safety can be shown by clinical studies other than confirmatory clinical trials. The slide shows the anti-cancer drugs that have been approved under conditional early approval system in Japan. So next, I would like to introduce the use of registry data in the approval review of anti-cancer drugs in Japan. A phase 2 trial was conducted in Japan in patients with HER2-positive colorectal cancer, which account for about 3% of all patients with colorectal cancer. Considering the difficulty of conducting comparative studies, registry data was used as the control group in this phase 2 study. So finally, I would like to mention Japan's approach to multi-regional clinical trials, MRCTs. It is important to consider conducting MRCTs as much as possible in order to create clear evidence and deliver effective anti-cancer drugs to patients with rare cancers. Therefore, attempts are being made by the Japanese regulatory authorities to facilitate the implementation of MRCTs. For example, the establishment PMDA Asia Training Center and so on. Finally, I would like to talk about the future challenge in order to promote the development of innovative anti-cancer drugs for rare cancers. First, it is important to promote harmonization among regulatory authorities and strengthen the system for conducting MRCTs. Second, it is also important to involve academia, as it is difficult for companies to create evidence for all cancers on their own. And third, it is also important to accumulating of knowledge and experience, then fostering a trusting relationship among related parties. So that's all. Thank you for kind attention.
Okay. Good morning, everyone. It's now my pleasure to introduce Dr. Sandra Kasich from the FDA. She's the acting team leader for the gastrointestinal malignancy team in Division of Oncology. She trained in medicine and in pediatric hemonc in Buenos Aires, Argentina. She practiced there and was also an investigator on international trials with the Cooperative Group for Pediatric Oncology. She joined the FDA in 2008 and has been part of the gastrointestinal team since 2013. Besides her review work, Dr. Kasich is engaged in the development of regulatory frameworks for the review of drug safety, developments of biosimilars, drug targeting pediatric cancers, and regulatory research projects. So it's my pleasure to have Dr. Kasich here.
Good morning, and thank you for inviting me to participate in this panel. When we were talking about the topics today, as you already heard, the important thing was to focus on innovative approaches to kind of overcome the development, the challenges of the development in rare diseases. However, in my mind, I needed to go through what we have been going through and know exactly how the framework that we currently are going in order to kind of know where we are, if the strategies are working, and how to move forward.
Can we get Dr. Kasich's slides up, please? Thank you.
Sorry, I have a slide that it's kind of heavy on data, and I think it's going to be better if we can show it before I continue talking. If not, I can keep going. But
Can we just have a thumbs up at the back if you're finding it? Okay. Not finding it?
Yep, absolutely. Okay. No problem.
Okay. So my here.
Just saved in time.
Okay. Getting your steps.
I like drama, apparently. Okay, I went through this. Or not. It's.
Next slide, please.
Okay, I have no disclosures, and also these chapters represent my views and should not be constructed as FDA views or policies. Here we are. So this is a snapshot of the approvals for drugs and biologics in the period from November 2022 to October 2023. There were 41 approvals, including 14 new molecular entities and 27 supplemental approvals for previously approved drugs. That means new indications or expanding indications to new populations for drugs that previously were approved. So when looking at endpoints and trial designs used for approvals of new molecular entities, 9 drugs were actually approved under the accelerated approval pathway, and they were all based on single-arm trials using overall response rate as a primary endpoint. However, of the 5 new drugs with traditional approvals, there were also 2 based on single-arm trials using overall response rate, And there were 3 approvals based on randomized controlled trials using time to event endpoints. For supplemental approvals, all those approved under the accelerated approval pathway were supported by single-arm trials using overall response rate as the primary endpoint. And of interest, some of the traditional approvals also were based on overall response rate, mostly assessed in single-arm trials, although there were 2 randomized trials that used overall response rate to support approval. In other words, 50% of all approvals in this period were based on trials demonstrating an effect on overall response rate, including 13 drugs approved under the accelerated approval pathway, for which further verification of benefit and additional safety data will be required. Importantly, about almost 30% of the regular approvals in the period were based on overall response rate, including some in very rare diseases. Atezolizumab was approved for alveolar soft part sarcoma. It was based on a 49-patient trial. Bosutinib was approved based on a 34-patient cohort of children with Philadelphia-positive chronic myeloid leukemia. Also remarkably in this period, there were 3 new indications based on randomized trials that were conducted in rare subpopulations of larger clinical trials, and for those, the statistical plan was designed in such a way that this analysis was prospectively planned. But there were also randomized trials in rare subpopulations. So some examples include the approval of dabrafenib and trametinib for pediatric patients with BRAF V600E mutated low-grade gliomas, a trial that enrolled 110 patients. The PROPEL trial in patients with BRCA-mutated prostate cancer enrolled 95 patients, and of those, only 47 received the combination olaparib and abiraterone. And in the RUBY trial, there was a subset of 122 patients with dMMR endometrial cancer. So based on these data, and acknowledging that innovation is needed to accelerate development in rare cancers, one may also infer when adequately employed, the currently strategies are actually working. So for rare cancers, and when appropriate and applicable based on mechanism of action, as our colleague just showed us, overall response rate is the most common and important endpoint used for approval, and is not generally interpreted in a vacuum. We always look at secondary endpoints that are necessary to further characterize benefits, and that includes duration of response, because short-lived responses probably are not that of benefit. But there are other endpoints that can also help characterize the benefit in terms of how patients function or feel. And examples of those are, for example, the registration trials of ruxolitinib in myelofibrosis, in which patients not only experienced meaningful reduction in spleen volume, but they also had improved symptoms scores. And there are other secondary endpoints. Examples are in prostate cancer that address also time to pain progression and delayed or prevented skeletal fractures and events. As mentioned several times before, randomization is always preferable when feasible, when there's equipoise, because randomized controlled trials are not only optimal to assess the treatment effect, but they also confer a lot of information regarding safety across arms. And there are several examples of successful randomized trials that were conducted in patients with rare cancers. I previously mentioned the approval of dabrafenib and trametinib for the treatment of children with BRAF V600 mutated low-grade gliomas. This was a small randomized trial, 2-to-1, between dabrafenib, trametinib, and standard of care with chemo. The trial enrolled 110 patients. And so for you to have an idea, this is a disease where there are about 1,600 new patients a year, but only about 300 of those have the particular mutation. You are all very aware of the CLARITY study that was also mentioned by Dr. Foggy. And there are approximately 720 patients, 750 patients a year in the US diagnosed with this mutation that have cholangiocarcinoma. So There was also mentioned previously, we are all familiar with the concept of tissue agnostic. This is a relatively new concept. The first tissue agnostic indication was approved in 2017. And we are familiar with baskets and molecular-driven designs like umbrellas and other that provide great opportunities, not only to leverage the already existing trial structure, but also to include these rare subpopulations for which standalone trials are unlikely to be conducted. So the approvals of some of these drugs that included patients with biliary tract cancer and cholangiocarcinoma, of course, provided new opportunities and expanded the landscape of these patients. And again, we have now these drugs approved that can be accessed by patients with cholangiocarcinoma, like dabrafenib and trametinib, Pembro, dostarlimab, and the new kidney block approved two weeks ago. That is trastuzumab deruxtecan for patients with HER2-positive tumors. So similarly for already approved drugs, there are opportunities to leverage data from prior approvals to support the conduct of smaller studies in rare subpopulations. Here you can see listed several indications for rare subpopulations based on really small single-arm trials using overall response rate as a primary endpoint for drugs with known safety profile and efficacy in other disease settings. Some examples include the approval of ivosidenib for the treatment of IDH1 mutated myelodysplastic syndrome. This is a trial that enrolled 18 patients. The approval of pemigatinib for the treatment of FGFR1 altered myeloid lymphoid neoplasm. This was a trial that enrolled 28 patients. And bosutinib for kids with Philadelphia-positive chronic myeloid leukemia. There are less than 150 new patients a year in the US. So finally, there are multiple other considerations. Some were alluded in the presentations before, and there are strategies to improve participation of patients into clinical trials for rare diseases. FDA has been working on initiatives and guidances addressing the centralized clinical trials, diversity initiatives, and expansion of eligibility criteria. The FDA Rare Disease Guidance provides advice for the use of auxiliary cohorts to augment the safety and efficacy database, including parallel cohorts. And in selected cases, it may include data from expanded access programs and real-world data. And there's also opportunities to design pragmatic trials, or at least incorporate pragmatic elements into clinical trials that will increase enrollment, given that patients will be able to participate closer to where they live and treated in a way that they would be treated in the real world. Also for investigators, collection of data is limited. So this is a snapshot of some of the initiatives of the Oncology Center of Excellence at FDA that are addressing interventions that directly affect clinical trials in rare diseases. Equity Silver and Asiatic are related to increasing enrollment of older patients and underrepresented minorities. Endpoint is a new initiative aiming to advance endpoint developments in oncology. There was an advisory committee last Friday discussing one of the new endpoints for multiple myeloma. Project Orbis is an ongoing initiative to facilitate the review of oncology marketing applications across participating countries. And we have multiple other projects. Those are available online. If not, you can contact me and learn more about this. This is a limited list, but there are more of selected FDA guidances and documents to help development of drugs in rare cancers. And last, I would like to acknowledge my colleagues who helped me with their insight, Drs. Barón, Emmerich, Klutz, and Donna Rivera. And above everything else, I want to thank you and the Cholangiocarcinoma Foundation for your attention and for inviting me today. Thank you very much.
And now my pleasure to introduce my Canadian colleague, Dr. Duc Vu. He is a pharmacologist with 30 years' experience in drug approval activities with Health Canada. He has been the director of several drug approval bureaus, His current position is Head of Oncology Division at the Pharmaceutical Drug Directorate of Health Canada. Dr. Vu.
Okay. First of all, I would like to thank the organizing committee for the opportunity to talk at the very inspiring convention. I really love every day that I spent over the last 2 days. Really, informative, and I learned a lot from every of the participants here. As the last speaker of this today's session, I think that it's a very, at the same time, easy and challenging. This is easy because a lot of things that I would like to talk already been covered by my, colleagues who just talked before me. And then challenging is how I make my talk a bit more informative so that I can highlight something that have not been talked before, and I would like to bring that to your attention. So my talk today is about the Health Canada regulatory drug approval processes and pathway for drug, especially drug to be used in cholangiocarcinoma. So I don't need to go too much into that. This said that 30% of the patients which have surgical resection that is the most profitable outcome that you can have. For unresectable metastatic patients, we have chemotherapy and targeted molecular therapies. This slide shows that the NCCN guideline recommends some molecular testing for targetable mutations that can offer some opportunity for targetable therapies. One thing I want to draw some of your attention is that, it has been discussed before, not all of those mutations are rare. Some mutations are less rare than the other. IDH mutation, FGFR, HER2 seem to be more common than other mutations. And that can dictate the way for us to think about clinical trial, how it can be conducted. And then, of course, for rarer mutations, we have to talk about more innovative approach to get the drug out. So, how can I approve? You all know durvalumab, gemcitabine, and cisplatin now become the standard of care. We approved that based on the TOPAZ phase 3 randomized trial with OS as the primary endpoint, OS benefits. The comparator is gemcitabine, cisplatin. On its own have OS benefit as well. And the evidence was from another phase 3 trial, ABC02. The primary endpoint here is also OS, overall survival. So those standard of care really come with solid evidence, phase 3 and overall survival as endpoint. Moving to more less common and rarer type of conditions, we are talking about CCA with FGFR2 fusion or rearrangement. Our colleagues who just talked before me discussed really much about pemigatinib. We know that those drugs have been available with the evidence of less robust evidence. We have, a single-arm trial. In Canada, they are approved under the conditional approval. I will talk about that a bit later. The primary endpoint used in those studies are ORR. And of course, all the drugs for rare disease that we approve is larotrectinib and entrectinib for the NTRK fusion. And those drugs get the agnostic indication for the metastatic tumors having the NTRK fusion, including CCA. And again, based on single-arm trial and primary endpoints is ORR. Dr. Fogi also talked about the IDH1 TKI, ivosidenib, It is under review in Canada for 2 indications, the AML and the cholangiocarcinoma for patients with IDH1 mutation. Again, to say that, the evidence to support that indication is a phase 3 comparative trial. Although the comparator is placebo, but at least there is some data to compare with. The key primary endpoint is PFS, which show again in the PFS, and the OS is secondary endpoint with some numerical advantage over the control. So now I will take a bit of time to talk about the approval pathways. In Canada, we have 3 approval pathways. The first one is the regular pathway, which will take 300 days to complete, and that covers the new drug submission and supplemental drug submission. The 2 other pathways are accelerated review, which is the priority review and the conditional approval. For the priority review, this is for unmet medical condition, drug for unmet medical condition, where there is substantial evidence for the clinical effectiveness. What we consider as substantial evidence is well-designed comparative trial with some robust endpoints like, PFS or preferably OS. The review time frame is much shorter than the regular time frame, which is now 180 days. The other accelerated pathway is the conditional approval, which is the notice of compliance with conditions. This is intended for drug, again, for unmet medical condition, but with promising evidence. What we call promising evidence is, anything else than randomized trial, phase 1 trial, phase 2 trial, single-arm trial, and the endpoint is, of course, not PFS and OS. With this type of approval, we need to have a confirmatory trial to confirm the benefits. We are not talking about only randomized trial as confirmatory trial. And we heard from our colleagues, from China, from EMA, and from FDA, any innovative approaches can be considered. Basket trial, umbrella trials, use of real-world evidence, etc. Those can be considered in the context of a confirmatory trial as well. In addition to the regulatory pathways that we have, we also have 2 collaborative projects with international agencies, which can bring to a more accelerated review time than the regular review time frame. The first international collaborative project is OCBIS. I'm glad that, that Dr. Kazak did not talk much about that, so I can have a bit of time to talk about that. This is an initiative put on by the FDA Center of Oncology Center of Excellence, with participation from several countries, including Canada. The review time frame is set by the FDA, which is very aggressive, and sometimes it can be shorter than our priority review. And of course, drug qualify for that OCBIS international collaborative project is drug for unmet medical condition, most likely falling within the review of the cholangiocarcinoma indication. The other collaborative pathway is Access Consortium, with several countries, including Australia, Singapore, Switzerland, United Kingdom, and Canada. This is a true collaborative project in the sense that each of the countries take part of one portion of the submission and review them, and then at the end, we consolidate the data. That can lead into some gain in efficiency, and again in the review time. But so far, we haven't used that pathway into the review of the CCA submission yet. Well, that slide is to say that we don't have a framework drug for rare disease in Canada, but we have a national strategy that was announced last year that the government will put some money, 1.5 billion over 3 years, to increase some capability to collect data from some health information agency like CAIHI, to have some collaboration alignment between the drug approval at Health Canada and the funding agency CADAT, etc. So those initiatives will be implemented in the near future. And my last point is to say, and to repeat some of the things my colleagues have talked about, points to consider in the pathway for approval. RCT is still the gold standard. Of course, if it comes with a randomized trial, solid endpoint, that is what we would like to have. Certain rare disease can be qualified for accelerated review, conditional approval, but those will need to have confirmatory trial to go with to support the evidence. One example I can bring here is one of the FGFR inhibitors, infigratinib, that we approved in 2021. We used the ORBIS type A approach, and then we gave the notice of compliance with conditional approval, and the confirmatory trial that needs to be submitted is a phase 3 trial compared with gemcitabine cisplatin. And that concludes my presentation. Thank you very much.
Thank you very much, and I'm personally very impressed to hear some of the initiatives that you're all bringing to try and accelerate the new drugs to patients. We're going to go straight into a discussion. So there's a couple of people I'm going to invite to the stage, Dr. Mitesh Borad and Dr. Teresa Macarulla. These are people who have both been introduced to you before, so they don't need any further introduction. And they're joining the panel as active clinical trialists in the US and Europe, respectively. Please disregard some of the rearrangement that's going to happen behind in terms of additional chairs. I then want to hand over to our next co-chairs. Wendy Seelig brings more than 25 years of experience in defining and implementing winning collaborative strategies, working at the top levels of government and within the leadership of the non-profit health sector. Wendy is known for connecting the dots for multiple stakeholders towards win-win collaborations that deliver tangible results. She fosters lasting relationships with senior federal government officials, industry executives, and healthcare non-profit leaders. Wendy brings a deep strategic understanding and the practical experience to get things done. And joining her is Jeff Allen. Jeff serves as the president and CEO of Friends of Cancer Research. For over 25 years, Friends has created unique scientific partnerships, accelerated policy change, and supported groundbreaking research to deliver new strategies to patients quickly and safely. He's a thought leader, and we welcome both of these chairs to take us through a round table. Dr. Knox and I will remain on the panel, if that's okay with you guys.
That's more than okay. So thanks, everybody. Sorry about running right through your break, but feel free to take one as needed. But we wanted to make sure that we had as much time as possible. It's a very unique circumstance that's been created here with all of these different regulatory leaders from around the world and all the stakeholders in the room, the patients, the caregivers, the advocates, the clinicians, everybody here together. I think we should take as much advantage as we can. Of all the things that I get to do, one of my favorites is to work with Stacy and her whole team and Juan and the Cholangiocarcinoma Foundation. I love what's happening here. I love this meeting, and I'm just happy to play a small part in this discussion. So we wanted to start with our clinicians who are up here, and maybe Juan, we'll start with you, and then Jennifer, and then we'll move on down the line to the end to our newly joining members here, Dr. Borad and Dr. Macarulla, sorry. I'd just like to hear from you guys to start. What's your reaction? I was feverishly taking notes, and our panelists from around the world, there's a lot of similarities of thought. There's a lot of innovation happening. How can we take advantage of that? How can the academic and clinician community take advantage of that to bring forward new treatments in cholangiocarcinoma? So Juan, maybe we'll start with you.
Yeah, so I'm going to be careful not to steal all the ideas because I'm sure there's many along the line. But I think a couple of things just that struck me from Dr. Chen's presentation was the run-through from the exploratory right through to the validation stage of a study, the confirmatory stage of the study, as a planned approach, rather than having a stop and start and stop and start, having that as a run-through. You know, we can see how that would be efficient and how that would accelerate. And I think the second thing I want to highlight that I took away is the increasing reference now to using real-world data with the caveat it has to be good data. And I think we as clinicians need to think, how do we make that data fit for purpose? But increasingly incorporating this into our development plan of drugs right from the get-go, rather than having to resort to the real-world data after the event. So it is something that I think we need to start building into our prospective plans. I'm going to pause there. I'm sure there's plenty of other ideas.
Thank you.
I would echo what Juan says. I was very impressed, actually, by all the different innovation that I saw across the different regulatory bodies around the world. And so I think there, you know, with some of these ideas of harmonization with different countries and these platforms where we get a control arm with or really good quality real-world data, it's all great. I do want to just, despite all this innovation, I do just want to point out that this conditional, you know, approval, which is fabulous as it gets the drug to market faster. You know, one example of where this may be a problem is with the FGFR inhibitors. If we can't get those large first-line trials done, you know, when you look at the history of how many drugs have actually been withdrawn because the confirmatory trial did not show the data, it's not very many, but there's just been a few. So I just wonder if when it comes down to the regulatory folks, if you could comment on, are we, you know, if a trial cannot get done in this rare space, what's going to happen to a drug that then has real-world experience and seems to be quite active? So that was one of my concerns, despite all the innovation.
So hold that thought. I see that the regulators are chomping at the bit to respond. That's a great question. So make a note that you wanted to respond, and we'll come back to that. But I do want to hear from our two down at the end, so from ENSCA and also from the ICRN perspective. Thank you.
Yes, so I completely agree with the question that once the drug is conditionally approved and it's in the market, it's nearly impossible to have randomized data. It's so difficult. And now what we have to do with these trials that are open and we cannot enroll, and what happened now. No? So I think, and more are coming. So unfortunately, that's something it's something that worries me. And I think the real world data is the key. So I completely agree with that. And then another thing that worries me, and maybe Dr. Foggy, because I am from Europe, no, it's the time. Of course, FDA is an example. It's fantastic. But why in Europe, and as I hear today in the other parts of the world, it takes this time to have an approval of the drug? And then in Europe, maybe Dr. Foggy can give us also the opinion, what happened, why in the different countries in Europe we have to wait until 100 days and more than 2 years in have the reimbursement, and our patients cannot be treated, and unfortunately, cholangiocarcinoma patients cannot wait 2 years to have access to the drugs. No? So why this time, and how we can improve it? So thank you.
Great. Okay, Mitesh?
Yeah, that was a very delightful and informative session, and I was really able to appreciate that all the regulatory agencies are very much patient-centric. It's not really an us versus them approach. They are really trying to help within their own legal constraints. A few things came up that were of interest. Defining what is clinical benefit has always been very ambiguous and vague, at least from my own standpoint when I viewed these things. And there's never been any defined metrics. How can you know how far you need to go if you don't even know the goalpost. And it's all been subjective as, durable, but, durable is an adjective. It's not a defined number. What should be the response rate? And I know the regulatory agencies don't want to box themselves in a corner later, but it would be very helpful to know exactly what constitutes success and correspondingly what constitutes failure. So we definitely want them to comment on that if possible. Also, patients have never really been engaged in defining what clinical benefit is. Everybody's asked everybody else except those people to whom it might matter. And you can see this is a room full of patients here, and I'm pretty sure most of them would be willing to give their opinions, and these are all intelligent, articulate people who could tell you what they feel clinical benefit constitutes. So just some thoughts for our regulatory colleagues to comment on.
Okay, great. Let's go back. I'm going to turn it to my good friend Jeff here in a second to ask some more questions of the regulators, but let's go back to the questions that Jennifer, that you raised, and that Teresa, you raised. So one is about what to do with the confirmatory studies in a rare population, and the FGFR was the example, and then the other was the time. And we're still joined by Professor Fogarty virtually, so hopefully you can still hear us, so please feel free to wave at us or jump in. So Dr. Kosaka, I think you grabbed the microphone when Jennifer was talking, so if you wanted to start.
Okay, so which one is
should be on.
Here. Yeah. So I think that on top of considering how rare a disease is, sometimes the conduct of randomized trials is very difficult because of the competitive space we are dealing with. And FGFR2 inhibitors is a typical example of that. Yeah, and rare and competitive space. So I think that once it was acknowledged that randomized controlled trials, at least the typical ones, cannot be conducted, and bear in mind that confirmation of benefit may not need to be on the same line where it was approved, so it can be conducted in a different line, which opens the space a little bit. There are other venues to confirm benefits. So if you look at the 3 products that have been approved for cholangiocarcinoma, FGFR-altered tumors, in the first one, there was a randomized controlled trial required. By the second and third ones, the requirement was for dosing studies, because sometimes these developments are very fast, and there was not enough optimization of the dose. So what we required are studies for dose optimization with response rate included in those. So it's a different type of randomized study. It's between doses. It requires less patients, and at the end, the goal is not only to verify the benefit, but also potentially offer patients the same benefit with less toxicities and a much more tolerable profile. So that is one possible venue.
Yep, go ahead. Dr. Wu, it should be on.
Yeah, to add into what Dr. Kazak just said, what happens if the confirmatory trial cannot happen or fail? The FDA recently have some thought that they would, before to give out a conditional approval, they would need to have evidence that, or proof that the company can be able to put in place a confirmatory trial. So that is the first thing that they would consider. They would ask if there is a good likelihood that such trial would be happening. And in the situation where confirmatory trial fail, they can consider another line of treatment. They can consider other approach, more innovative approach. Doesn't need to be, a randomized, comparative trial. This can be real-world evidence, as long as the protocol is solid and sound, with all of the control for biases and so on. We can probably consider that. So that is, the option that can open to the drug industry. Yeah.
Maybe just to follow up on that. a question to all of the global regulators on the, I think each of you have mentioned the role of real-world evidence. Have your agencies utilized real-world evidence as confirmatory studies in the past, and what would be the limitations of doing so currently?
Not yet. Currently, not yet. Yes, we are thinking about that, the real-world data to support the registry. But currently, I think the data quality may be the important, crucial question, because currently we only use the RWE to support the indication, the supplementary. Yeah. So.
Yeah, I would agree. I think that at this point we have not used any data to as the only data set that would be confirmatory or even for approval. It's always supplementary and complementary, and it all depends on the quality of the data. The FDA guidance also talks about parallel cohorts and expanded access programs, which may also complement limited data sets from clinical trials.
Dr. Foggy, did you have a response?
Yeah, no, I guess my response is overall in line with the response of the colleagues. What I can tell you more is that we are trying to work hard to liaise with the academic world and the registry holders, because I guess that what we really need, and I guess that Dr. Valle, Professor Valle alluded to that, it's really something that we need to engage each other. And we have a very interesting meeting a few weeks ago at EMA that was about the registries and the qualification of the registries in order to be usable from the regulatory perspective. And then what came out clearly is what we are kind of speaking different languages, even in the semantics, or what we expected. I mean, even the most valuable academics and registry holders really struggle to understand what our needs are. And perhaps, I would say the other way around, perhaps we were not so good in explaining what we need in terms of regulatory expectation in order to enter the era in which we can really use more and more this real-world data, because I guess that's where we could make the difference. So we have a process in place to qualify these registries. We have a qualification advice, and then qualification opinion. We have qualified some registries, and we had some experience in starting to use this registry from a regulatory perspective, but it's a way to go. But yeah, and if I can say also on the, don't stop me for talking too long, sorry, but just I wanted to address the other issues that were, on the conditional and the confirmatory trials. It's the issue ongoing on different lines. We used several times. It's tricky because we had also experiences in which the trial failed in a different line. What to do with that? And for instance, you have noticed in pemigatinib there was a mix on providing the final results of the trial and the phase 3. And then we are struggling. Who knows? I stop here. But sometimes we use replication. I guess we did so for the products in the agnostic indication or tumor-independent indication, and we request the replication on phase 1 trials. That's more feasible, maybe, although many uncertainties could remain. So in rare disease, it's always challenging. In less rare disease, perhaps we should consider then looking at early at ORR in the same trial, in which is going to look at a time to event endpoint might be a way forward. Then we have struggling with the integrity of the trial issues that might turn out to be a problem, but perhaps it can be handled and it can be a way forward. Access around Europe, it's painful. I just can tell you that we as regulators try to liaise as much as we can with the HTAs. And now you know that at start of 2025, the new HTA regulation is coming in place. The mean joint scientific consultation and joint scientific assessment, and hopefully, I cannot promise anything, but the hope is that it will reduce the time to real access, because as I always say to my colleagues, after the CHMP opinion, nothing has happened for patients if the drug is not in the market. And this is a crucial issue, and thanks for putting forward.
Right. Thank you. Maybe one follow-up to our clinical colleagues. I think you've seen an openness for the utilization of real-world data, but a challenge in terms of data quality. In terms of opportunities for improved collection of data, what are some of the challenges that are encountered? And I was struck in Dr. Kosak's slide, she mentioned increased initiatives at the FDA for incorporation of pragmatic elements into clinical trials, and I think at least in the initial prototype in lung cancer, we've seen a great deal of success where an initial pragmatic trial has been able to enroll patients significantly faster than a typical clinical trial. In fact, I think the study was planned to be about 4 years in duration, and after year one, it's already well over halfway accrued. So I guess the question to the clinical experts here, what are the challenges in collecting this data in the context of regular practice?
Yeah, for me, one of the challenges is to find the patients, because we are talking about a small number of patients. So, for example, HER2 amplified patients or specific, I don't know, if we want to be RAF patients. So I think that collaboration is absolutely crucial, no? Not only in a country, also in the different countries worldwide, in order to collect this data, and of course to do it properly, guided by people who are specialists on that, with good CRF, with resources to do it, because otherwise, if the physician, after to visit the patient, have to put the information there, probably it's not a good quality, so the resources also are important.
I think we also need to have clarity on what are the key data to collect. We know from some of the retrospective studies that we've done in our centers that capturing toxicity data, for example, is very poor. Capturing some of the harder endpoints in terms of timing of scans and responses and so on is less challenging. So I think one of the challenges that we have is knowing in the real world setting what is essential, that has to be very good quality. I know it all has to be good quality, but are there some data fields, for example, capturing lots of grading of toxicities, starting and stopping and concomitant medication? That's a very big burden. Is that also required to that degree?
I think that's exactly the goal of the pragmatic trials. Collection of data is really limited. If you look at pragmatic lung, there's no collection of overall response rate or PFS data. It's only survival. Toxicity is limited to grade 3, 4s, and deaths. So it is really there. But the big caveat of that is that it needs to be in the setting of a very well-known drug for which the toxicity profile is known. It's not something that you can do with a new molecular entity.
Dr. Foggy.
Dr. Foggy, you said?
Yes, go ahead.
Okay, sorry. Yeah, no, I just wanted to say that it depends on the context of use that you're targeting for your registries. I mean, or for your collection of real-world data. And And of course, the type of granularity and the typing in the way data can be captured that can make the difference in the usability for different regulatory output. So it's the context of use. So I guess that, and then this can be expanded or can be tailored. And there are various uses that you can do. And depending on the tools that you have and the granularity and the quality and the interoperability and a lot of stuff, there are things that are important. But yeah, so this is the kind of way of interaction that we try to have and exchange with clinicians, acknowledging that there are different, very different situations, but especially in rare disease, I guess that cooperation and trying to try then to design some studies within the registry perhaps is something that could be done and well thought in advance for specific contexts of use.
Thank you. I wanted to broaden the discussion a little bit and talk about innovative trial designs. I think all of our regulatory friends referenced openness to platform studies, basket studies, umbrella studies, adaptive design, which is encouraging, and we've seen publications from the FDA as well encouraging that, and I think everybody here knows that there is a move afoot to bring that kind of approach into the cholangiocarcinoma space, and Jeff and his organization have been involved, and I'd love to maybe start with Mitesh and talk a little bit about, the opportunity, but also if there are questions. We want to make this global, right? And so everybody generally seems favorable, but maybe the devil might be in the details a little bit, and we'd like to make sure that there is harmony before we get stuck down a blind alley, so to speak. So maybe, Mitesh, you talk a little bit about that.
Yeah, no, thank you for bringing that up. Conceptually, trial designs where you only set up a trial once and then keep it open and keep amending and innovating it so that you remove a lot of the inefficiencies of starting new projects every time seems like it's an ideal solution. I guess the details are in the background, which is when you have this kind of setup, there needs to be trust between the academic partners and the sponsors from biopharma, if they are the sponsors. They could, of course, be governmental sponsors or other bodies. And with it would come a little bit of a loss of control in terms of trusting that the collaborators will carry out the trial in exactly the manner that was intended. And I suppose that it's this perception of loss of control which diminishes the enthusiasm at times for partaking in master protocols or basket studies, which would otherwise be extremely efficient.
Yeah, I completely agree. So cholangiocarcinoma is a perfect setting to do it in a small percentage of patients, HER2 amplify, BRAF, and we can demonstrate the activity. It's fantastic, but then it's true that in some countries, for example in Spain, that's completely impossible to have approval with this kind of design. So it's fine. I know that FDA and EMA approves some of them, but then when it's question of the different countries, it's very, very difficult to have the reimbursement in this cohort of 40 patients in this basket trial, and in fact we didn't have any approval for this kind of drugs. So that's something that have to change, or maybe we can, I don't know, uniform a little bit, or at least in Europe, the new rules that are coming.
So is there something that the patient advocacy community can do to enhance that acceptability? And then Jennifer, I saw you wanted to make a comment as well.
So they have a powerful that we don't have, so absolutely they can help a lot with this in the different countries, in the country level, for sure, because they have the power to do it. We don't have, unfortunately, the power to do it.
I was just going to add that, we heard a lot about, access to drugs in different countries and delays in that, and so in a way, this kind of a platform study would be particularly appealing, internationally, because we're motivated to find the patients, we're motivated if we sequence them and that to get them on something and then keep them on a platform and collect the data. My concern would always be, do I have the control arm funded, or is it available? So these are the kinds of things that we're talking about when we're trying to plan it, to make sure that it is something that answers a question but is also applicable globally. I would say I don't know of any barriers sort of in my country to do a trial like that, other than just the fact that it takes commitment and time and effort. But I think once it gets going, I think it's actually going to be a win-win for patients, for investigators, and for the sponsors.
And maybe if some of our regulatory colleagues would want to just comment a little bit. You all raised this, so what type of experience have you had with this? And do you see any barriers? Are there concerns coming from the sponsors that you're hearing? Are there concerns about the level of data that you're getting? Just love to hear a little bit more from you about some of these innovative designs that you've had experience with.
Yeah, maybe is it on? Yeah. We approve those and track fusion with the commitment that they follow to provide more information via platform trial. So so far we haven't heard any negative things from the sponsor. So I presume that this has been ongoing, and yeah.
How about in China or Japan?
Actually, in China, most of the platform trial were initiated by the investigator. As I mentioned in my presentation, the Professor Wu Yilong, he built up a clinical network. We call it the CTON, which is in lung cancer and breast cancer. 10 years ago, they had already initiated such a platform study. They combined the different companies and with different target product in one trial. It's open trial. If there's a new compound, there's a new target, then they can add it. That yes, we. Not yet. Currently, we haven't approved a product based on platform trial, but I think it is under the way. Yeah, because you can select one of cohort to support. A single arm cohort to support one product to get approved.
Maybe just to follow up on a couple of kind of common threads that have been raised in a couple of contexts. So I think in each of your presentations, the majority of you at least describe programs that your agencies have put into place around providing early input with sponsors. Whether it be some sort of breakthrough designation. Or programs that are tailored toward development of drugs for rare oncology indications. I'm going to follow that with the description from the last presentation around Project Orbis and other global coordination activities. And it seems to me that I think in the description that you provided around Project Orbis and I will acknowledge the success that program has had. I think FDA has published that it has yielded access to drugs on the magnitude, in some cases, of numerous months, if not close to a year or more, faster in certain circumstances by having that coordination. But oftentimes, I believe that's around the review process of the ultimate application. Is there any similar opportunity to move the elements of Project Orbis earlier in sort of the study design? Because I think we heard issues around predictability, consistency around global requirements. So could there be an opportunity for global coordination at that early sort of breakthrough stage where some of the studies are still being designed, given that many of these studies are global endeavors?
Yes, there has been an FDA and EMA program for years. It was used a lot initially with the biosimilars, in which there is a common meeting with all regulatory agencies and a common path. But I think that besides Orbis, we do have a very strong collaboration with EMA in particular, Canada, and PMDA, and other stakeholders. And these are discussions that we have all the time. We have monthly meetings. Issues that the regulatory frameworks are different, so I don't think that total harmonization is possible. But definitely collaboration in early stages would help on that. And those are happening, maybe not as organically as one may wish in the current environment, but those are happening.
And maybe from the investigator side, is that
it was actually a follow-on question regarding the harmonization. Professor Nonaka talked about harmonization. Obviously, in the early stages, the data is the data. We may only have a single phase 2 study with a good overall response rate. There is not very much else to submit. Is there something towards harmonizing what the submission dossier needs to look like across various agencies, so that there is a single point of submitting all the data together that then gets... I completely accept that the evaluation may differ by country, but maybe the papers, the requirement for the information that's submitted maybe should be standard.
I think it is mostly standard. The difference is that we receive data sets and we do analyze those. Most agencies don't. I think it is about to happen in Europe too. But as of now, we are the only ones reviewing raw data. But we follow the international standards. Sponsors submit the same packages all over the world.
In China, we already joined the ICH, so the ICH guideline, we also follow that. So I think the international, we also accept the ECTD, so it's almost the same.
I wonder, Dr. Foggy, if a couple of times in your presentation, when you were talking about the two examples, you talked about crossover as part of the trial design, and sort of maybe with little asterisks, and even though crossover happened and so forth. I know, and if Melinda's anywhere in this room, that the opportunity for crossover in trials is extremely important as patients are reviewing whether or not they want to be part of a clinical trial, and having the best shot at accessing the investigational therapy. And this is especially true where, the standard of care leaves something to be desired. So I wonder if you could talk a little bit more, maybe you start us off, it's sort of the perspective on crossover and what the caveats are, and then I would like to hear a little bit from our other regulatory colleagues and from our physician colleagues as well about how that type of a design element weighs in here.
Yeah, of course we know that sometimes is the only way to get trial done. From the pure assessment perspective, as you can imagine, crossover is not what we want to have. Then we try to handle that. But let's be honest, there are methodologies also to correct for crossover. They're all based on assumptions. And so we will look at that as sensitivity analysis. Then on the primary analysis, we go for the ITT principles. Then we have the estimate framework, and and we look at the interquartite events, and we try to handle that. But yeah, in a way, it might be sometimes a case by case the evaluation also at the time of the planning stage of the design. But yeah, we try to handle, but mainly the ITT principle is what we try to look at as the primary analysis. And when things go fine, yeah, perhaps it's less as an issue. It becomes an issue when the the results are very borderline. Let's be honest on that, and that's the truth. Because it's a worst case that we try to handle in advance at the planning stage. Just to a short comment on the fact that at least from the clinical trial application perspective and the marketing authorization perspective, which is slightly different, but there is a clear openness to platform trials and master protocols and things like that. Then perhaps it might become, as usual, an issue when it comes to reimbursement, because we had approval based on platform trials. And of course, there is a lot of discussion when you use a common control arm, for instance, on the type 1 error control or things like that. There is you say the devil is in the details, but I just wanted to make it clear to the audience that in Europe there is not an issue with platform trials or things like that.
Thank you. Yeah, go ahead, Dr. Wu.
Yeah, crossover. This is individual fact of the clinical trial in the context, especially when the control is placebo. That would be unethical not to allow a crossover. But we know that when we deal with crossover, the data or overall survival might be totally not interpretable, especially in the context where you start to see, the favorable result. That would be difficult for us to interpret that. Although there are well-established correction adjustment statistical method to correct for the crossover, as Dr. Foki said, we use those correction as sensitive analysis only, and we base our assessment on the ITT, following ITT. So if we see a negative OS result after a crossover analysis, the question that we ask is probably, does the second line treatment would be better than the first line, for example? We have to ask the hard question, should we really approve a drug where after crossover you see a gain of, a detrimental OS data, then you have to engage in the conversation with the sponsor to see what makes sense from the result. So that is all I can say.
One strategy maybe to avoid bias, especially if the trial is an open label trial, is essentially to avoid informative censoring, is to consider real-time blind and independent review of progression and response. So in that way, that is objective and the patients were not dropped because they felt that the other arm was better.
Yeah. Well, agree.
Any other comments from the regulators? Otherwise, I'm curious from the clinician's perspective, and I'm going to turn it back to Jeff here. And in case you're wondering about the schedule, we've decided to go a little longer because we want to take advantage, and we're going to break at 1:15. So from the clinician perspective, with regard to the patient view of the design and crossover, curious about your thoughts.
I can comment. I wouldn't want to go backwards. I think the crossover, as we move into this era of precision medicine, and we don't want to increase the disparities with access. As a clinician speaking to a patient who has a druggable mutation that needs more data, I would feel comfortable providing it to them in either a first or a second line setting, depending on the situation of the drug with that crossover. So I take the point of having the primary analysis blinded, so that it's because we are very biased when we believe in a new drug or something. So I think that is a very good point and something that we need to think about, but then allowing the crossover. It's going to happen sometimes in the real world anyway, right? As people get access to drugs in different ways. So I think we need to plan it in. I think that it is appealing when I have that conversation with a patient about that kind of, I think they understand it. You have to present to them that there is a standard of care that we don't want to throw out too quickly, but there is an innovative idea, and you're either going to get it first or second, or depending on the context. So I think that appeals to clinicians and patients much, and if we can get the quality of the data out, then that's great.
Yeah, just a quick comment on the crossover. This has come along a few times. Of course, it did in the ivosidenib trial design, and even trial design for an ongoing study. When there's a placebo control, it would basically be unethical to not allow a crossover. And just because we want to see beautiful overall survival curves, they come at the expense of the demise of patients, and that is just unacceptable from so many standpoints. So I would urge patients, investigators, advocacy to discourage patients from participating in trial designs that are geared primarily to help the sponsor or get a clean signal. That's really not ethical at the end of the day.
In Dr. Kosak's presentation, she mentioned a program at the FDA looking at unique and novel endpoints, including the discussion that was had about a week ago around a molecular-based endpoint of minimal residual disease in multiple myeloma. I'm curious if the other regulatory authorities have any similar programs that are not necessarily drug-specific, but geared toward validation of earlier endpoints. And is that anything that is of interest within the clinical community in this particular space?
Yeah, maybe, for example, ctDNA being explored as a response metric. I mean, RECIST is kind of old and not as objective as we'd like.
Trials are ongoing. We'll see. Hopefully it works.
I'll take that as an openness for exploring additional endpoints. Or Dr. Foggy?
Yeah, we also are open to exploring other early endpoint. MRD for sure is one that is under discussion and intensive discussion and in different settings. But yeah, it's science that is evolving, and the regulators try to take the pace of science without lowering the requirement, but with open mind. I guess it's a usual way forward, and so we are there. Yeah.
Great. Well, I think we will wrap up. I'm going to turn things over to Juan for a couple of closing comments, but I should have prefaced our start with this, I think, but I want to thank particularly our regulatory colleagues that have joined us today. Oftentimes, your important role is found in the crosshairs, where perhaps there's a good portion of the population that believes that everything is done too fast and the other half believes that everything is done too slow. But we certainly appreciate your expertise, your willingness to have these discussions, and for all of your efforts. And thanks for being here.
Just to echo Jeff's comments, I think the drugs wouldn't be available without the regulatory authorities working in the background, making sure that drugs are safe, making sure they're effective. So thank you for the work that you do. Thank you for taking the time to come and spend some time in this round table. I'm sure that the conversation is hugely appreciated by patients and caregivers in the room, as well as investigators and everybody. So thank you. We'll take a break.
Thank you. Bye.